हाय हेलो एवरीवन। वेलकम टू माय यूट्यूब चैनल जिसका नाम है नॉलेज हब। अगर आपको वीडियो पसंद आए तो लाइक शेयर सब्सक्राइब करना ना भूलें। आज का हमारा टॉपिक है इंसुलिन। सो व्हाट इज़ इंसुलिन? इंसुलिन क्या है? एज यू कैन सी इन दिस डायग्राम, इंसुलिन इज़ नथिंग बट इंजेक्शन। एज यू कैन सी हियर in this on this bottle what they have wrote, injection. So what is insulin actually? Insulin are even made from the pancreas of pigs and cows. How do we extract insulin? Insulin is extracted from the pancreas of animals such as pigs and cows. Insulin is even extracted from the bacteria, even from the human pancreas, and is even made commercially. So, insulin is a naturally occurring hormone made by our pancreas. Our pancreas means human pancreas, that helps our body use sugar for energy. If your pancreas doesn't work as it should, it may not make or release the insulin. You need to control your blood sugar, resulting in the case of diabetes. It is mainly taken by the patients of diabetes. As you can see here, these are the bottles of insulin. Insulin is a polypeptide hormone produced by the beta cells of the islets of Langerhans. Its main function is enabling the cells to take up glucose, means providing it with energy it's need. So mainly glucose, main function of glucose is to provide energy. So providing it with energy it needs. It requires for the normal glucose homeostasis. What is it? For the normal glucose homeostasis. What is homeostasis? Homeostasis means level, means to maintain the normal glucose level. So this is what the structure of human insulin is. As you can see here, chain A, chain B and chain C in this diagram. Chain A is a alpha chain B beta chain, and chain C, as you can see here in the gray color, it's an additional chain which is called as C peptide. How do we study the structure of insulin? Insulin is composed of two peptide chains, as you have seen in that diagram, referred to as alpha chain and beta chain. Alpha chain and beta chains are linked together by two disulfide bonds, and an additional disulfide is formed with a chain. Alpha chain consists of 21 amino acids and the beta chain consists of 30 amino acids. Next, let us study the importance of insulin. What are the importance of insulin? Why is insulin important? Without insulin, the blood glucose builds up in the body and the cells are starved for their energy source. What are the symptoms of diabetes mellitus? The symptoms of diabetes are fatigue, constant infections, blurred eyesight, numbness, tingling in the hands or legs, increased thirst, and slowed the healings of cuts. When this happens for too long a time, the body produces ketones. What are ketones? Ketones mean it is the one which measures or the level of your urine. So this is why ketones is produced in the case of diabetes. Chemical production, in the case of chemical production, and even it is produced by the liver. The ketones are mainly what we call it as, it's mainly an acid which is produced during diabetes. So this is what a diagram showing human insulin. It's an injection. We have your insulin pen. This is a needle. Here we have needle attachment point. This part what we have got, we call it as insulin reservoir. It's a cap of the injection here. This is what we call as insulin cartridge. This is an expiration level, means date of expiry. Next is dose adjustment dial. This is what here we have in the red color. This is an injection button. As you can see here, how do we measure diabetes? This is what measuring it is. This is the syringe injection. This is the bottle of insulin. Next we can see how insulin is produced using recombinant technology, which means rDNA technology. The gene responsible for producing human insulin protein is isolated. A plasmid is isolated from a bacterial cell. Special restriction enzymes are used to cut the plasmid. The gene for insulin is inserted into the plasmid. Where is it inserted? It is inserted into the plasmid. This process is called as recombinant technology. We call it as rDNA technology. The bacterial DNA now contains the human insulin gene and is inserted into the bacteria. When bacterial cells will reproduce, the human insulin gene is also reproduced in the cells. Where is it reproduced? It is reproduced in the cell, and it produces the protein by the process called as transcription and translations. So here, this is a diagram where scientists build the human insulin gene in the laboratory. So this is how the human insulin gene is prepared in the laboratory, where DNA has been inserted, and then they remove a loop of bacterial DNA known as plasmid. As you can see in this diagram here, plasmid where the loop of bacterial DNA has been removed, and then it results in the bacteria. Here how bacteria is removed. This is what the bacteria, removed bacteria. In this diagram, we can see that the human insulin gene is inserted into the plasmid. Here we have human insulin gene where DNA has been present and it has been inserted. The plasmid, this is what the human insulin gene into the plasmid has been inserted. As you can see in this diagram, this is the plasmid where the human insulin gene is being inserted. In this diagram, we can see that researchers returns the plasmid into the bacteria, and later it results in recombinant bacteria has been released here. So this is what human insulin gene, as you can see, the yellow color, where the what we call it as plasmid is being returned to the bacteria. So this is what we call it as recombinant bacteria. Here what they have done, we are returning the plasmid. This is what the plasmid we have here. We are returning it to the recombinant bacteria. In this diagram, what we can see a fermenter tank and the recombinant bacterium here. We are putting the recombinant bacteria in the large fermentation tank. This is what recombinant bacteria in the green color. So this we are inserting in the fermentation tank. Next, in this diagram what we are doing, the recombinant bacteria use the gene to begin producing human insulin. Using the fermentation tank, human insulin has been produced. This is what the recombinant bacteria, putting it into the fermentation tank, and then what is the result? Recombinant bacteria will produce the insulin here. Here we have fermentation tank where the scientist will harvest the insulin from the bacteria, and then the insulin has been harvested here. This is the fermentation tank where the recombinant bacterium will produce the insulin. Here insulin has been harvested. At last, we will purify the substance for use as medicine for the people. So this is what we have: insulin, the purified insulin we are harvesting. After this, after purifying, after harvesting and all, at last we'll purify it, and then what becomes? It becomes a medicine. So guys, thank you for today. If you like the video, don't forget to subscribe, share, like and comment on my videos. That's all for today. Thank you.